Bismillahirrahmanirrahim. So today we are going to start our chapter 5, and in this lecture we are going to see what is the understanding of the attachment of the viruses to the cells and how do they enter the cells. All right. So first of all, you need to know that viruses, they are of different sizes. There are some big viruses, some small viruses, but all of them, they need to have some types of programs, some types of gadgets to enter the cells, right? They simply cannot diffuse across the cell membrane because these are the obligate intracellular parasites, and they must get inside of a host cell in order to reproduce or in order to increase their number, but they are way big, so they can't mm, even if a big virus or a small virus, even just the nucleic acid like RNA or DNA, they cannot go inside the cell through the cell membrane, and they cannot diffuse across the plasma membrane. So they have to have some specific mechanisms, and that we are talking about today. They need to attach to some kind of a molecule that is present on the surface of the cell membrane, and that molecule, my dear, is called as receptors. Here you see. So receptors, how to define receptors? These are the signal receiving molecules. Whatever signal uh, given by the organism, by the nervous system, or by the endocrine system, or by the feedback mechanisms, these are the molecules that will receive a signal and process it and send the message to the interior of the cell, and cell will respond accordingly. These receptors, keep this concept in mind that these receptors are not there for viruses. These receptors are there for the normal physiological functions of the cell, but the viruses, they evolve themselves to be able to get into the cell, and receptor, sometimes it is more than one protein involved. Sometimes just one receptor is involved. Sometimes we need two more co-receptors for the viruses, that the viruses can go inside the cell. So the plasma membrane, sometimes within the endocytic pathway, sometimes we need endosome for the viruses, and sometimes the viruses, they need to have and access to the nucleus. Sometimes they need to have access on the cytoplasm. So that's why we will study that what are different strategies that are employed by the viruses to get attachment and to get inside the cell. So here you see the receptors, they are the key to the cellular communications, and as I told you, there are many receptors in our body. Just our sensory mechanisms, you know that we have lots of senses, five senses, and then these senses are they work as per our nervous system, and they receive the signals and they process those signals, and the target cells, they respond accordingly. So that's why it is very important to understand about the chemistry involved in the receptor ligand bindings, receptor viruses binding, and we will see that what are different types that are present here. Here you see that these are the bonding molecules. These binding molecules, they are called as the ligands, right? And these ligands, mostly they are of protein, and receptors, they are also most of the time they are protein, but they may be of some other macromolecules like the carbohydrates, right? So these macromolecules, they will have some kind of biochemical reactions. Most of the time, as I told you, that they are protein in nature. So there has to be a protein-protein interactions, and proteins are made up of amino acids, and a receptor may have different domains and ligand binding sites on its receptors, and that will make the viruses unable to get themselves attached, and then they can go inside the cell. right? So there are some steps for that. We have three steps for that. The virus has to find the right cell. As I told you, that receptors are specific. All the cells are not able to take in all the viruses. For example, hepatitis C virus will go into the hepatocyte. It will never go into the kidney cells. It will not go into the heart cells because the receptors over there are not present. And those cells that not having the proper receptors, the viruses will not go inside those host cells. And in the previous lectures, as I told you that those plant viruses or the bacteriophages, they are not going to attack the human cells because human cells lack the receptors for their binding. So that's why the plant viruses, they will just come inside our body and they will leave our body without doing anything. So that's why we need a proper receptor for proper infection, right? So these three steps, the as I told you that the specific receptor is needed for the entry into the cell, and sometimes more receptors are needed. Sometimes just a single receptor is enough, like HIV just uh, is the example of having two receptors, and hepatitis C virus is also having one or two co-receptors. And similarly, the receptor for the coronavirus, the new studies have shown that there are also some co-receptors for the main receptor is ACE2, but there are still some co-receptors that help and assist the main receptor for proper binding and entry into the cells. And then the final step is that the transfer of the genome inside the cell. That's the most important thing. This is the genome which is protected by the capsid outside the cell. So it is the most precious part of the virus. So this genome should go inside the host cell, otherwise it will be useless, right? And these are there these some examples are there that the server receptors are present for the viruses, but there are some viruses that don't need any kind of receptors, right? And like fungi, they have no extracellular phase. These viruses that are causing infections in the fungi, so fungi will not have any kind of receptor. So these receptors are present with the fungi throughout life. So as the fungal cell is going to divide, the virus will also divide and it will transfer from generation to generation. And also another concept that we discussed in previous classes as well, that the plants as they have a cell wall, so it is very difficult to break the cell wall. And most of the viruses, all the viruses, they will go either through the plant injury, or there is another method by which the viruses can go inside the plants, and that are the insects, right? And those insects can cause infection to the plants. And there is another method by which viruses go inside the plant cell, is that whenever the plant machinery is used, you know, the tractors and trolleys and cultivate for the cultivation processes, it is used when some crop is cultivated or when some sowing is there. So at that point, some of the plants, they are damaged, and then the viruses, they have the opportunity that they can go inside the plant cells. So the receptors, this one is the flu virus. If you can remember it, it has 8 segmented molecules, and in 1985, we just know one receptor, and that was the sialic acid, and that was for the flu virus. So this is relatively a new field of investigation, and since 1985, we have discovered a lot of receptors, and now we have lots of technologies, lots of advanced techniques. Like for example, for coronaviruses, we just have discovered the virus in December 9, 2019, and just after a few weeks, the receptor was known, that which ACE2 was the receptor responsible for the entry of the coronaviruses into the cells, right? So these days the technology is very fast, and this is also important about the vaccine development. Normally, vaccine takes 8 to 10 years, but within after the we just have almost 11 months to the infection of the coronaviruses, and the vaccine is going to be available very soon. So things have been changed over time. So how you are going to study the receptors? First of all, the recombinant DNA technology is used, and then we have monoclonal antibodies, flow cytometry, nucleotide sequencing, and small interfering RNA and CRISPR-Cas9. Okay, so recombinant DNA technology, how you are going to utilize the recombinant DNA technology? You just isolate the genes and put it in a plasmid, get the protein, get the sequence, and see what is the product of that DNA. And then you go for the 2D or 3D structure. Then you have the amino acid sequences. You can go for different docking analysis. You can go for the properties of the amino acids present in that particular protein that is encoded by the gene. So recombinant DNA technology can also be very helpful to study the receptors. And then we have monoclonal antibodies. Monoclonal antibodies are the antibodies that we use to block the viruses, right? Sometimes when there is a viral infection, and that viral infection has caused the production of antibodies in our body, and those antibodies are also having the memory in them. And whenever there is a second infection, those antibodies are going to block the antigens present on the surface of the viruses. So that's why we have flu all the time, right? But the flu virus keeps changing and coming back to us as a new strain, as a new kind of virus. So that's why our body is not going to fight with it, and our memory cells, our antibodies that already are present will be useless if we are infected by a new virus, or we are infected by a new strain of the flu virus. But still, those monoclonal antibodies present in our body, they can give us a clue that what type of receptor or what type of protein is important for the entry of the viruses, and for the specific receptors present on our cells. The flow cytometry means that you are going to study the cells one by one. You have millions of cells, for example, here, and you will label them with particular dyes, and then you have a laser light through it, and the laser light will detect a particular fluorophore that already being labeled onto the cells, and it will tell us that what type of cells are there, whether they have a particular receptors on their surface or not. And then we have nucleotide sequencing. Nucleotide sequencing let us determine the sequence of these receptors. And then two more recent technologies we have: a small interfering RNA and CRISPR-Cas that allows to disrupt a gene uh, that you can knock out a gene, you can knock down a gene. And the CRISPR is also a gene editing technique that you can, if you are studying a receptor, you are suspecting that this is the receptor involved, then you take the cell, and you took the gene out of it, the gene responsible for encoding that particular receptor, and the cell receptors for the viruses will not be present then, right? If you are removing a particular gene from a cell, from cell's DNA, then that receptor will not be present on the surface of the cells. And then you can control, then you can examine, then you can then you can analyze the comparative analysis of both the host cells, one with the gene and one without the gene of the receptors. So the example that I will quote here is about the same example in which the CCR5 gene of the embryonic cell was removed by a Chinese scientist, and that CCR5 is responsible for the attachment of the HIV particle, and it serves as a receptor for human immunodeficiency virus. So using the CRISPR-Cas technique, that particular scientist has removed that gene. When the gene is removed, the receptor is gone. And when the receptor is gone, HIV cannot attach and enter into the cell. So these are different technologies that are being used right now in the field of virology to study the receptors. Right. So criteria for identifying the cell receptor by the viruses. So these criteria means that what are the different points that we should look for the identification of the receptor, because it is not very easy. Whenever we have a new virus, then it is not easy that what receptor that particular virus is using to get entry into the cell, right? So for that purpose, we should have a clinical data as well, and we should have a kind of all the information and how the receptor is going to bind with the virus particles. And sometimes what we do, we just isolate some of the receptors and we isolate the viruses, and then we mix them up in a particular media. And if in the solution the virus particle and the receptors they bind with each other, we can identify that which receptor is going to bind with which virus, right? And similarly, the antibody to the receptors, it can block the infection. So these processes mostly they are performed outside the body. So we have the receptor gene, we have the viral genes, and we can both these molecules we can study outside the cell. And if they have affinity with each other, if they have I told you that they have self assembly process. Every protein has a self assembled information buried in their sequence. So by default, they will fold in a particular shape, and this default will lead to a position where the proteins can interact with each other, right? So receptor and ligand, they will have a conformational match with each other, like a lock and key. A particular receptor will bind a particular ligand. A particular ligand is going to bind with a particular receptor. This is a kind of a lock and key relationship between the receptor and the ligand. Okay, so these are the techniques, these are the criteria that we use for the study of the receptors for the viruses. As I told you that the cell receptors, we have such a huge number of receptors in our body. Only our nose has trillions of different orders. Our nose can sense trillions of orders. Human nose has 400 different types of receptors, and it can distinguish at least 1 trillion different orders. And this is just about the nose. And what about the taste? What about the other sensory parts of the body? You have lots of billions of receptors are there, and they can have lots of sensing. But again, these receptors are for our own body function, but the viruses, they are smart enough to get through these gates. And for example, here you see that these viruses, they are going to use the HSPG protein present on the cell. Here, these are the attachment factor. This is the sialic acid for the flu, and these are the low density lipoprotein receptor. These are for HCV and HRV2. And similarly, for HIV, we have, as I told you, we have two receptors, CCR5, CD4, and CCR4 is also a co-receptor. So these receptors, actually, they have their own functions. The most of the time they are protein in nature, but carbohydrate molecules, some lipid molecules, some mixture of carbohydrate and lipids, some glycoproteins, they are present on the cell and serve as the receptors. Most of the receptors they are present on the cell membrane, but there are some reports that some of the receptors they are also present within our cells, and those receptors are known as the cytosolic receptors, right? So the receptors they have attachment factors, and the viruses here you can see the many viruses can use the same receptor. Here you see the IAV, measles, RSV, RV, HCV, HRV2, and here Ebola viruses and DV, UUK. These are the different viral infections. This is FMD, HC, rotaviruses, and many viruses can use the same receptor as a gate for their entry. Okay. Here, so different viruses can bind the same receptors, as I told you here. So adenoviruses and Coxsackie virus, they have a common primary receptor, and the swine herpes virus, pseudo rabies virus bind the same receptors as the human polio virus receptor, known as PVR, polio virus receptor. And then there is another aspect. That aspect is that one virus can bind to different receptors. And the herpes simplex virus, as I told you, that it is the virus that can never uh, leave human being alone. It is always there right from the beginning till the death of the individual. And this virus has why? Because it has lots of receptors present, and these receptors are present on almost most of the cell types: HSPG, integrin, nectin, HVM. So that's why these viruses are going to attach with almost every receptors, and they can go. For example, one cell has more HSPG receptors and don't have these receptors, these integrin, nectin, HVEM, OSHS. So the viruses will just use this gate to get inside the cell. And if integrin is abundant on a particular cell type, then this HSV is going to use this integrin gate. And similarly, these gates will be utilized present on the cell. So that's why herpes is forever. As I told you in the lecture one, that unlike love, herpes is forever. So how the cells allow the viruses to go inside them. So here you see the cells basically are very rushy area. Normally, whenever we see a photograph of a cell, we see just lots of the empty spaces. But these spaces actually are not empty. This is full of cytoskeleton. It is full of ribosomes. Just a single cell have millions of ribosomes in the cell. And we have endoplasmic reticulum, we have Golgi apparatus, we have mitochondria, nucleus, and some other molecules freely floating in our cells, and the web of cytoskeleton, the microfilaments, the microtubules, the intermediate filaments, they are all over there. And then we have a cell membrane. It is made up of lipid bilayer, so it is not very easy to get inside the cell, right? And that's why they just cannot simply diffuse into the cell membrane. They cannot diffuse into the cell. They cannot just cross the membrane. They can never do it, the viruses. So we have phagocytosis, and we have endocytosis. These are the processes by which the viruses get inside the cells. The phagocytosis, it is normally for the bigger viral particles. And here you see after attachment with the particular and specific receptor, the cell will take the particle inside the cell. And here you see the macropinocytosis and the receptor mediated endocytosis. We have a ligand here, and this ligand can bind with the receptor, and then this whole the vesicle can go inside the cell. And then after vesicle formation, the genome can be released there. So these are different methods by which the viruses are going inside the cells. So another important factor is the fusion of the viral and host cell membranes. As I told you that the enveloped viruses, they have the membrane that we call as a viral envelope. That viral envelope basically it is derived from the host membrane, right? So it is very easy for the enveloped viruses to get attached and fuse with the host cell. While the same facility is not available for the non-enveloped viruses. In the coming slides, we will see that how non-enveloped viruses they go inside the cell. It is a bit difficult for them, but yet they go inside the cell and cause infection. Like the rotaviruses, like the polio viruses, they don't have any kind of envelope, but still they cause infections. And here we see the clathrin dependent fusion and we have clathrin and caveolin independent fusion as well. Here you see the microfilaments, they are present uh, nearby the cell membrane. Here cell membrane is lipid bilayer, so it is not very easy to go through these webs of the microfilaments. Then we have microtubules here. These are the microtubules, and even during this process a virus has a particular pathway. It is not like that they are just propelled into the cell and they are just thrown into the cell and they will find it by themselves. There is a proper highway, proper motorway, proper roads for the viruses. They will travel on those roads. They will follow those protocols and SOPs to reach their destination. Sometimes they need to go inside the nucleus here through the kinesin protein. Kinesin protein, we will see it is a motor-like protein responsible for the movement of the virus particles along the microtubules here. And here you see the genome is going inside the nucleus. And here some of the genomes they are not needed by the nucleus, so they will just uh, stay within the cytoplasm. So these are some molecules, clathrin dependent here. These are the molecules that are needed, and dynamine, it is a kind of structure that is made by these molecules here, right? And then these are the clathrin molecules, and the clathrin coated vesicle is formed, and after that we have an endosome. So endosome basically it is a structure that has acidic properties. Sometimes this acidic property is going to release, is going to unstable these capsid molecules and unstable these nucleocapsids, so that the main player is the nucleic acid. So the main player can go out of that shell and can perform its replication or transcription or translation activities, whatever needed. Okay. So there are caveolin dependent and caveolin coated vesicles. There are clathrin coated vesicles, and sometimes like the flu virus, they don't need clathrin or caveolin. They just go inside through the fusion of their own particular membranes. So these viral fusion proteins, they are present, these viral membranes basically present, viral fusion proteins are present in the viral envelope. Okay, so this is very important to understand that these proteins are present on the viral envelope, and these proteins are going to have a collaboration with the proteins present on the surface of the cell membrane, and then they will go inside the host cell. So how the uncoating of the viruses is done in the host cell? This is the most important process, because here the nucleic acid which is going to be released after the attachment. Here you see the HIV virus, and the HIV virus is going to have a kind of fusion here with the membrane, and then after fusion this particle is released, and this particle will then be moving towards the nucleus and will be integrated into the genome. And then we have influenza viruses that will go into the cytoplasm, and after the cytoplasm they will make an endosome, and this endosome will unstabilize the structure of the nucleocapsid, and the capsid and these nucleic acid, the RNA molecules, they will go in the cytoplasm first, but they have a nucleolar signal, so they will go move again into the nucleus for their caps. And then we have these are the viruses not having envelopes of polioviruses. It will attach with the particular receptors, and then it will make a vesicle, and it will be endocytosed within the cells here. And when it is making an endosome, this endosome is having a low pH, and this low pH will destabilize the is going to destabilize the viral product. Sometimes the pH is not necessary; just the attachment of the receptor with the particular ligand present within the poliovirus. So we will see that what types of ligands are present within the particles that are not having envelope, how they get into the cells. They have on their hexagonal or here on their hexagonal or pentagonal surface, they have some kind of fibers, and these fibers they get detected by these receptors, and then the instability of the capsid occurs, and this instability will lead to the release of the genome inside the cell. And then we have adenoviruses. Adenoviruses again, they are having fibers. You see here, these are the vertical fibers. They are present here. This one, so they are recognized by the membrane proteins, and these then there is endosome formation, and endosome then there is a lysis, and again uncoating near the nucleus, and then the adenoviruses can go inside the nucleus, adenoviral genomes. Okay, so the main concept is that there are proteins, there are fusion proteins present on the viruses, and there are some fibers present on the naked viruses, non-enveloped viruses. They are going to form some endosomic structures, and then after the destabilization of their capsid, they will find their destination inside the cell for replication, for transcription, and for translation, and later on for their assembly and release. So just one example, this is this sialic acid. Sialic acid, this was the first ever receptor discovered, and this receptor is a carbohydrate, and it is for receptor for the influenza virus. Here you see the sialic acid. It has this is the structure of the sialic acid, and neuraminidase is here. You can see, and this particular bonding is is shared by the birds as well as by the human beings. So that's why sometimes we have viral infections from the birds, and birds can have viral infection from from the human beings, right? And the main thing is that here you can see the receptor for the influenza virus. There is a proper fusion of the membranes. Here you see, for example, this is the cell membrane here, this is the cell membrane, and this is the virus being endocytosed through receptor mediated endocytosis, and the endosome is here. After the endosome is made, and this genomes, the capsids and the viral envelope that is present on the around these nucleic acids, it should be destabilized, and there should be a kind of release of the genome, because if the genomes are not released, then the virus will not be able to replicate. So here the proper fusion of the proteins that occur, and this fusion will lead to the instability of the particles, and that will result in the release of the um, genomes, and they will go through the nucleopore inside the nucleus for their proper um, transcription or replication, and they are going to utilize that machinery for that. Okay, so what are different types of fusion proteins? These are the fusion proteins present in the viral envelope. We have two classes, and normally we have three classes, but we are going to discuss two classes here. The class 3 is the complex one. For the complex viruses, the class 1 fusion proteins, they are perpendicular to the membrane, just like spikes. We have spike, like in coronaviruses, we have spikes in the MERS, we have coronaviruses, and these spikes are helicals. They are mostly of helical nature, that alpha helices protein we have, and they always made the trimers. And here we have influenza viruses, semen viruses, Ebola viruses, HIV, and murine leukemia virus, and we have coronaviruses. These viruses are more active. Keep that in mind, that if a virus is having a class 1 protein, and because of the alpha helical structure, they will be having more opportunity to penetrate inside the cell membrane rather than the class 2 fusion peptides. Because the class 2 fusion peptides, they are mostly, here we see, they are mostly beta plated sheets, and they form dimers, and they are parallel to the membrane. So that's why they will have less opportunity to go inside the cells. They can go, they will go, but the viruses having class 1 fusion protein, they will have more opportunity, they will have more facility for the fusion. And this fusion is always regulated. It is it is it should not occur in the wrong location, otherwise there will be no fusion. Most of the time, as I told you, that all the viruses, they are not ideal viruses. They have deformities, they have a kind of you can say disabilities, because fusion is also a factor in that. Sometimes there is a fusion at the wrong location, so those viruses will not be able to infect. Okay, and there is protein interactions during the fusion, and then the pH also plays its role. And the fusion of the endosome membrane is also very important. Sometimes, endosome has a membrane, and this membrane is going to fuse with the receptors as well. So it will play a part. The fusion is going, the regulation of the fusion proteins, whether it is a class 1 or class 2, they are going to play a part in the in the entry of the enveloped viruses. But now the question is that how this is the way how the enveloped viruses are going inside the cell. How the non-enveloped viruses, they will go inside cell. So these are the genomes released by the non-enveloped viruses. Here I told you that they are going to have a kind of fiber here. So these are the adenoviruses. They have a fiber, and they have a fiber receptor for that, and after binding with their particular receptors, again here you see the endocytosis is being done, and these are the uh, clathrin coated bits we have in this cell, and this endosome is formed here, you see. And this endosome, again you see it has receptors here, because this endosome is also made from this membrane, the cell membrane, and it has this integrin, and this integrin is going to destabilize this in the capsid, and the capsid is destabilized, and because of the acidic environment here, the genome is released, right? So this genome is released in just within an hour. The nucleic acid is inside the nucleic acid of the adenovirus is inside the nucleus, where it will perform replication and transcription, right? So, and they will travel through microtubules. As I told you that the destination of the viruses, the destination of the nucleic acid, it's not random. It is highly specialized. For example, if we want to go from Lahore to Islamabad via motorway, so there is a proper plan. There is a proper road for it. Okay. So similarly, the viruses have a proper road, a proper highway and motorway, that where they are going to and how they are going to found that destination. Okay, it's not a kind of random movement. It is a particular planned movement for the viruses that they are going to find their destinations. So this is again an example of the poliovirus. Here we have adenoviruses. They don't have any envelope, and they go inside through receptor mediated endocytosis, and then the endosome, and then the acidic environment destabilizing the capsid, and then the genome is released. Similarly, the polio virus is there. Polio virus is having a particular polio virus receptor. CD155 is the receptor for the polio virus. It is present in our intestinal epithelial cells, and 90% of the people having polio virus in their intestine, then they will never know that they have a polio virus in their intestine, because it doesn't do anything mostly in the intestine. So whenever this polio virus migrates towards the brain area, then there is a problem, and then it can cause the disability of our lower limb. Okay, so here you see that these are the cytoskeleton part here. This is the receptor, and after the receptor is attached, the particle loses the free energy, and this after this energy is lowered, then the particles become the destabilized, and the poliovirus is released through the pore in the endosome membrane formed by the VP4. This viral protein 4 will make a pore in the endosome, and then this uh, pore is also used for the release of the poliovirus genome. So another example is the rotaviruses. These are very important viruses, and 600,000 deaths per year in the world occur. It is and the size of this virus is 75 nanometers, and the capsid its capsid is made up of 3 proteins layers, and the genome having 11 segments of double-stranded RNA. Wow! And this particular virus is providing us another information here, that how this virus is going to attach with the particular receptors present on the cell membrane, and then endosome is formed, and then the proteolysis, and then the lysosome is fused with the endosome. This is the infectious subviral particle present here, and as I told you that it has 3 layers. So the layers are dissolved by the lysosomal acidic environment, and then the penetration of different types of molecules, it will go inside the viral particles. Okay, so sometimes the ribosome will go inside the virus particle here, and uh, mm, they are going to have a kind of protein synthesis. But there are some reports that because it has a double-stranded RNA structure, whenever it is exposed in the cytoplasm will have the mechanism of killing any nucleic acid present in the cytoplasm. Okay, cytoplasm has that protective mechanism. Our cell has that protective mechanism. If, for example, any DNA or RNA other than messenger RNA and ribosomal RNA, if some other type of nucleic acid is present, like the double-stranded DNA is present, like the double-stranded RNA is present, then our cell is going to oh, recognize it as a foreign antigen, and it will immediately try to cover it up and will kill it. But there is a process we will uh, learn in the coming slide, that how this double-stranded RNA uh, genome is used within the cytoplasm for the production of messenger RNA, because double-stranded RNA cannot produce proteins as such. So this double-stranded RNA will be having an mRNA synthesis within the particle, and then this mRNA will be released, and this ribosome will read this mRNA, and then the process of translation and then the assembly of the viruses will take place. All right, so these are the 4 modes of nuclear entry. Here is the flu virus using the nuclear pore, and then we have herpes viruses. Sometimes the caps complete capsid is attaching to the external part of the nuclear membrane, and then the virus is released here. And this is the adenoviruses, and this is the parvoviruses, very small viruses, and sometimes they can go inside the nucleus to form the replication or transcription processes. So this was the lecture about the receptors and how the cells they are attaching the viruses, and I have some uh, videos for you that how they are going to get inside the cell. So first we have the entry of the coronaviruses, how the coronaviruses they are going inside the coronavirus. So here you see this. Okay, these are the cells waiting for the corona. These are the coronaviruses, and these are the spike proteins here. So virus genetic material is present. This is the capsid, and these are the proteins that are present outside. This is the envelope, and then we have capsid, and inside this capsid there is nucleic acid. Okay, so the envelope. This is envelope having these protein spikes, and these protein spikes are going to find their respective receptors. ACE2 receptor will be found by these spike proteins, right? And after finding their particular cell type, they will have a fusion. Okay, these the viruses are released in the droplets, and these droplets are traveled and finding a new host. And here is the new host, and they get inside the body, and inside the body we have throat and nose cells, and these cells are having a lot of ACE2 receptors. And here comes the receptor, this one, and this receptor will take this virus inside the cell, and this cell membrane sac is having a kind of attachment with the nucleus here. Okay, and this after the destabilization of the virus particle, the nucleic acid is exposed to the ribosomes, and ribosomes can read that messenger RNA, and it can give more and more proteins, and then these more proteins they will be processed in the Golgi operators, and they will assemble again, and they will go outside the cell again. Okay, now they are going outside to find another host, another healthy cell. And we can see the another example here. We have this is the viral process of HIV. This is how the HIV is going inside